Good morning. I'm Richard Martin. You can hear from my accent that I grew up in the UK. I'm going to talk to you a little bit about what we do in our labs, and we've got a set of slides here that I'm using to talk about our lab. We focus on drugs and drug targets in nematode parasites, and particularly on ion channels. The title slide here, I've got my name here, Richard Martin, but it's important to recognise that I work very closely with a number of colleagues, particularly my long-standing colleague, Dr. Alan Robertson. We're in the Department of Biomedical Sciences at the College of Veterinary Medicine here at Iowa State. I said that we're a group of different colleagues, and here's a slide that shows us who we are. And because we're interested in nematode parasites that attack both animals and humans, the human aspect allows us to gain funding from the NIH, and the animal aspect brings us a commercial relevance that will allow us to investigate some of these different compounds that we're looking at. Here's Alan on the left of the PowerPoint slide, and here's myself here, and you can see quite a diverse bunch of young colleagues. We're a pretty exciting group, and if you want to know anything about international food, in addition to drugs and drug targets in parasitic worms, come to our lab. So let me introduce what we do and some of the diseases that we work on. And there's a picture here in the left-hand side of my screen of a lady who's got this rather unpleasant condition called elephantiasis. It's blocked drainage of the lymphatics due to a parasitic worm. It's transmitted by biting insects. It's rather an unpleasant disease, and if you've got this condition known as lymphatic filariasis or elephantiasis, then essentially you become socially isolated. Very unpleasant. But what's the connection with animals? You get a parallel system, a filarial infection, relatively common in the southeast of the US, Georgia, Louisiana, in the southeast of the US, very common condition. This heartworm condition, this filarial worm, transmitted by biting insects that blocks the pulmonary arteries of young dogs. They have to be treated regularly with a monthly antiparasitic drugs. That's the kind of condition we're interested in, both in humans and in animals. They're the same drugs. Look at these other conditions that we look at. Ascariasis in a human, very common, reckoned that at least a third of the human population is infected with these parasites due to lack of sanitation, preventive measures. This poor child here with this enlarged stomach, and look at these kinds of numbers of worms you can get out. Ascaris lumbricoides is said to be, some authors say, the same parasite that we get in hogs here in Iowa, Ascaris suum. Some people say they're genetically the same. We can get these parasites from the local packing plant. So a range of the diseases that we're interested in, nematode parasites. The kinds of drug that we work on, some examples of what we've worked on and published on. Imidepside, relatively commonly used for treatment of small animals, roundworm infections, now being considered by a variety of locations tropically for the prevention and control of filarial infections. We've worked with this drug, which was developed by originally Upjohn and then Pfizer, and it then morphed into Zoetis, Derquantel. We've worked with this and helped get it licensed. Some old drugs, diethylcarbamazine, used for the control of some filarial infections, lymphatic filariasis. We've discovered the new mode of action of this working on TRP channels. A couple of old drugs, levamisole, we find that it works in a more complicated manner than has been assumed. And this compound, oxantal, that works on whipworm, we found a specialist ion channel, ACR16, as a target for that. This compound, ivermectin, many people have heard about this. This resulted in the award to Bill Campbell of a Nobel Prize. We've worked with this drug and looked at the effects on glutamate-gated chloride channels. So some of the drugs, anthelmintics, they're known that we work on. So some actual parasites that we can get hold of in the lab. We've worked with this heartworm. We can occasionally get these from post-mortem dogs, and we've been able to conduct electrophysiology on these worms. Ascaris, I've shown you that they come from the hog. We can get that regularly from the packing plant. And this character supplied by the NIH FR3 in Georgia. This is Brugia malayi, the guy that causes elephantiasis. So those are some of the worms that we work on. Here's some of the targets. Remember, this is what we're talking about, drugs and drug targets. These are the ion channels, or examples of the ion channels that we work on. These are pores in cell membranes and quite complicated proteins. I'm not going to go into it, but they cross the cell membrane, and basically they open and close, and when they open they will allow different ions to flow through. They'll cause currents to flow across the cell membranes, and we're able to record those fantastically small currents, sometimes down to the size of a picoamp. That's a millionth of an amp. We can record them as these ion channels open and close. So we worked on nicotinic channels, quite common, GABA channels, these are inhibitory channels, and these GluCLs, glutamate-gated chloride channels, that are opened by the drugs that relate to ivermectin. This little picture in the center of the diagram here is one that we're quite proud of, from a recent publication where we discovered the mode of action of this rather ancient drug, diethylcarbamazine. People originally thought it only worked on the immune system of the host. It doesn't. It also has an action on the parasite. And we've shown that it works on these rather new, interesting TRP channels. These are quite fascinating ion channels with peculiar properties. We've been able to study how drugs work on these ion channels when we've used the Brugia preparation. And here in this little diagram, in this slide, is where we've been able to work on the drug emodepside, and we've shown and produced evidence that it works on these rather interesting, what we call slow 1 potassium channels. These are great big channels that hyperpolarize, inhibit the muscle cells. Emodepside, this is the drug that's fairly commonly used in dogs and cats, and now being considered for use in humans to control filarial parasites. So some of the techniques that we use, what we do, some of the techniques we use are patch clamping and calcium imaging. Here's an illustration of our technical equipment that we use, an inverted microscope, fairly high resolution to see our cells, and there's a little picture in the middle. Can you see these single muscle cells of Brugia? Quite high resolution to see, and there's a diagram of a pipette, patch pipette, that we put on the surface of the muscle cell to pick up these currents when we open these ion channels. And this diagram here is one I particularly like. It fascinates me. You can see the actual molecular changes in structure as the ion channels open. You can see this current pulse here. The channel, the ion channel, is closed, and look, zonk, it opens, gets to this level, zonk, it closes. Here's a bigger ion channel, it's closed, look, zonk, it opens, stays open, then closed. The amplitude of this current is nearly 1/millionth of an amp. That's the kind of things that we're able to do. I think it's fascinating. This guy here and this diagram here, is we've got some calcium fluorescence we've injected a dye inside the muscle cell. We've treated this preparation with diethyl carbamazine, and it increases the calcium fluorescence. It's a bit of fun doing these experiments. They're not easy to do, but that when you get them to work, it's rather exciting. It's like catching salmon, if you like, going fishing. Perhaps that's an unfashionable word, but we do it nonetheless. How do we express some of these ion channels? We're actually able to clone these ion channels, because not all parasites are easy to get hold of. We're able to get hold of the genetic structure, copy them, and then actually we're able to inject them into these toad Xenopus oocytes. This is a diagram. Here's this little character up here, female. We collect the oocytes and we inject the cRNA and the oocyte. It makes the ion channel, and we can record the ion channel as it opens and closes. We do this again with this voltage clamp technique. It's a bit, I don't want to go into too much detail. One electrode for fixing the voltage and the other electrode for controlling the current as the ion channel opens and closes. Here's an example in this recording here. We put on a drug, levamisole. You can see how it produces this inward current pulse as it goes in here. And we can look at the different protein requirements, different components are required to get this ion channel to operate successfully. These ion channels are not just single creatures. They work together with a number of colleagues, if you like, accessory proteins that help proper behavior. That's what we study as well. Here's a little bit of screening that we've done, an example of some testing of a series of different analogues that we were able to get hold of, and some that we were able to synthesize with our colleagues in chemistry. And we were able to do this modeling and looked at a number of substituted pyridine derivatives, looking for improved, more potent compounds that were active against this ACR16 nicotinic receptor. We were hopeful of finding an improved agonist that might be useful for treating trichuris whipworm infections. So there's an example of some sorts of screening that we can do using that Xenopus oocyte experiment. The worminator. This is a phrase that seems to have caught hold of. And what is it? It's a sort of wiggleometer. You put these worms in various 12 or 24 well plates. You could also put them in a petri dish if you like, and I don't know whether you can see them on the screen, but there's a little wispy character here, and you can see there in the control. We have a camera on it, and these worms move and they wiggle around. And this, what used to be called a wiggleometer, but that didn't have the same cachet as a worminator. So here's the worminator going now. We're measuring the movement of this worm here. And when we put a drug on, basically here it becomes paralyzed. And we put on an example of a drug. Here we can see diethyl carbamazine. It inhibits the movement of the worm. This very simple observation here that we can see this inhibition was not expected and certainly wasn't predicted in the literature. we've shown that DEC has a direct effect on the parasite, and this was the starting observation. It turns out that this effect is relatively short-lasting. It only lasts for a couple of hours, and this explains the transient effect of diethylcarbamazine. So the Worminator then is one of the other gizmos that we use. Academics, university academics, promote themselves to some extent by their publications, and here's an example of the lists of, we think, pretty good recent publications that we've been able to demonstrate what we've been able to do. Some publications in the journal PLOS Pathogens, Nature Communications Biology, and Trends in Parasitology describe some of our recent studies in a little bit more detail. So that's some examples of our publications. What are we looking for? Why am I here? I think the simple reason we're looking to industry, we recognise that there's some significant questions that we can help to answer. We're looking for industry to help funding to match the support that we get from NIH. We're also looking, another very important aspect that we've found works well for us, we're looking for some novel compounds. The industrial chemistry and the med chems characters that are in industry come up with some very interesting compounds that are novel, that would be very interesting to test. So those are the two prime things that we're looking at. Matching funding, if we can get some from industry, and some novel compounds that we'd like to test. And with that, I'm going to close this slide. I'm here presenting the lab as a character, but look at these colleagues. This sort of productivity does not occur without significant support. I mentioned my colleague Alan Robertson, Sudhanva Kashyap, Shivani Chowdhury, Melanie Abongwa, Paul Williams, Charity Neo, Rial Datta, Mark McHugh, Saurabh Verma, Tina Hansen, Dan Clerk, Claude Charvet and Cedric Niveau. These are collaborators in France, in Tours, and Dan and Tina in Denmark. Look at the funding that we have needed to support this work. We've been able to get funding from NIAID, the Benbrook Foundation, and we appreciate some support, considerable support from Bayer, INRA and Iowa State University. And with that, there's a final picture here of the gentle doctor. We are obviously, as veterinarians, have a grounding in animal health, but very significantly, the animal health, when it comes to parasites, expands into human health, and it's a very good example of the One Health agenda. I'd like to thank you for your time.